Hey guys, this is going to be Unit 6. I've broken this down into two separate videos, the first of which is going to cover genetics discovered by Gregor Mendel. I do want to say, first of all, if you have not done your vocabulary or you have not started working on it, you need to do so as soon as possible. There are a lot of terms in this unit that are very important. And if you don't know your vocabulary, you're not going to be able to understand what the question is asking you. So vocabulary, very, very important. Please make sure you have done that and you are studying it. This video will cover Objective 5B, which is Mendel's laws to evaluate Punnett squares and different types of Mendel's laws. That's going to be dominance, incomplete dominance, codominance, sex linkage, and multiple alleles. So we'll cover all that in this video. First, let's take a look at plants. Plants reproduce through sexual reproduction, which means that they make sperm and eggs. You can see that picture of the flower on the left there. The stamen is what makes sperm, which is the pollen, and then the carpel produces the eggs. So normally that type of flower would self-pollinate, which means that although it is reproducing sexually because of the gametes, it's really sexual reproduction through one parent. The offspring are still genetically diverse because of crossing over when the gametes are formed, but there's technically only a single parent in self-pollinating plants. So Gregor Mendel was a monk. He is considered to be the father of modern genetics, and he experimented with pea plants. I'm sure you've heard this whole story before. The pea plants are traditionally self-pollinating. Mendel wanted to cross-pollinate them to track certain genetic traits through various generations. So the pea plants don't take up much room. They reproduce fairly quickly, and he was able to cut off the sex organs of those flowers and then cross-pollinate them based on the traits that he wanted to take a look at. So genetic traits are characteristics of an individual, and in this case with the pea plants, that would be seed coat, plant height, flower color, things like that. Traits normally vary between individual offspring in sexual reproduction because of crossing over during meiosis. So in order to track his generations of pea plants, he called the first generation the pea generation for parental, and then the consecutive generations were named F1, F2, F3. The F stands for filial, so F1 would be the first filial generation, F2 would be the second filial generation, and so on. So in the chart at the bottom of that page, you can see that those are the crosses that Mendel did. So we've got the pea generation there. So he crossed for seed shape, seed color, seed coat, pod shape, pod color, flower position, and plant height. So in his F1 generation, what he found was that instead of those traits kind of blending in to get a mixture, he saw only one specific trait was carried on. So for example, seed color. He crossed a yellow pea with a green pea, and all of his offspring were yellow. So in his crosses, he said the nature of one parent seemed to disappear in the first generation. So he was curious what happened to that trait. So he continued experimenting to find out if the other trait had disappeared or if it was still there hiding out. So he allowed the F1 generation to self-pollinate, and then he could look at the next generation to see if that trait would reappear. So in his F2 generation, the example given here is he crossed a tall plant with a short plant. So his F1 generation gave him all tall plants. Well, when he let those plants self-pollinate, in his F2 generation, he got a small percentage, roughly 1/4 of the next generation, produced short plants. So what he assumed was that being a tall plant was dominant, and that masked the recessive trait. So since the recessive trait reappeared in the next generation, it indicated that the trait for being short had separated from the trait for being tall. In other words, instead of getting all medium-sized plants back, he got either tall or short. There weren't any in between. So he came up with some different ideas. These can be called Mendel's laws or Mendel's principles, but these are based on his observations of his pea plants. So his principles are the principle of inheritance, the principle of dominance, the principle of segregation, and the principle of independent assortment. The principle of inheritance states that traits or biological characteristics are determined by individual units called genes. The traits are passed from parent to offspring. So remember when we talked about proteins in the last unit, we said that the proteins are controlled by genes, and genes are sections of the DNA that code for specific proteins. So the coding on your DNA codes for proteins, which is expressed in the traits. And these are those traits that are expressed, whether they're yellow or green seeds, whether the pod color is yellow or green, whether it's tall or short, those kinds of things. Those are the inherited characteristics that the offspring get from the parent. Then we have the principle of dominance. So that simply states that some traits are dominant, some are recessive. The dominant trait will always show if it is present. The recessive trait will show only when the dominant trait is not present. So you can see in that little picture on the left, I liken the dominant trait to a bully. So it's always going to bully or push out that recessive trait unless that dominant trait is not there. And that's the only time that the recessive trait is going to be allowed to show. Next, we have the principle of segregation. So adult organisms that reproduce sexually have 2 copies of each trait. And we talked about that a little bit in the last unit. So when the gametes form, the traits separate or segregate independently. So in that picture on the right, you see that would be the adult organism at the top with the matching homologous chromosomes. And then when the gametes are formed, either sperm or egg, those homologous chromosomes are separated. And then when the zygote is formed, those chromosomes can come back together. So remember, humans have 46 total chromosomes, but we have 23 pairs. So 23 from mom, 23 from dad. And then finally, we have the principle of independent assortment. So this just states that genes for different traits do not segregate or separate together. So for example, if Mendel was studying a cross between a yellow smooth seed and a green constricted seed, those traits aren't inherited together. They're actually separate. So there's a gene that codes for the color, and there's another gene that codes for the shape of the seed. So the Punnett square on the left would not be correct. The Punnett square on the right would be correct because that's going to show that those 2 traits are going to separate. They're not going to be together. They're going to segregate independently when the gametes are formed. Allele is one of your vocabulary words. So traits that are controlled by one gene that occur in 2 varieties. So remember, you're getting 1 chromosome from mom, 1 chromosome from dad. The gene variations are expressed in different forms. Those different forms are called alleles. So you can see in the picture at the bottom, we have 1 chromosome from mom, 1 chromosome from dad. That R that's in that red band there is the one that's going to code for the seed coat. So the capital R would be for smooth and the lowercase r would be for constricted. So those alleles are going to come back together, or those chromosomes are going to come back together when the zygote is formed. And that section of that chromosome is what's going to code for that seed coat. Segregation is another one of your vocabulary words. When gametes are produced, the alleles for each gene segregate. So each gamete contains only one allele for each gene. And if you look at that picture on the right, you can see that those two alleles are together at the top in those little purple circles. So that would be coding for tall plants. Then those alleles, those letters, are going to separate or segregate when the gametes are formed. And then they come back together in different forms in the offspring. Homozygous, another one of your vocabulary words. Remember, our prefix homo means the same. So this would be two identical alleles for the same gene. So in this case, it would be big T, or little t. So the resulting trait can be either dominant or recessive. And you can see in the picture to the side, that is I believe that's probably for eye color. So we have big B, or little b. That's homozygous. They're the same letters. They're matching letters. Then we have heterozygous. Remember, your prefix hetero means different, and that's going to be different alleles for the same gene. So that can be big T, or big B, little b. The resulting trait will show the dominant trait. Phenotype is the physical makeup, so it's what it looks like. And you can see there's different phenotypes at the bottom. So either smooth, or constricted for the P's. And I always associate the pH and phenotype with physical makeup. Genotype is the genetic makeup of an organism. So geno is equal to genetic. And that's going to be what the alleles are. So the B's or the T's or the S's, it's going to be how you're going to write it on the Punnett squares. That's the genotype. Probability is another one of your vocabulary words. It is the likelihood that a particular event will occur. So in genetics, probability is used to predict traits. That's not saying that your offspring will have these traits. It's just saying there's a possibility of these traits being expressed. So probability can either be expressed as a percentage or a ratio, and we'll get into that a little later. You can see in the Punnett square on the right, we have mom and dad are crossed. So they have a 25% chance of having a dark-haired child, 50% chance of having a brown-haired child, and then a 25% chance of having a red-haired child. We'll get into that in just a minute. So Punnett squares are what we use to predict the probability of the genotypes and phenotypes of offspring. There's another example of a Punnett square there. Now, I know you have made Punnett squares before, but let's just go over it. So first, you're going to write the genotype of the organisms that you're going to cross. So you need genotypes for both parents. In this example, we're going to cross a male and a female eagle that are heterozygous for large beaks. So heterozygous means they have the different alleles. So the big B means they have a large beak. The little b means they have a short beak. So since they're both heterozygous, you have big B, little b for both parents. So then remember, those segregate when the gametes are formed. So you're going to have big B and a little b. Then you want to draw the table. Make sure that it's as square as possible. And then you're going to write the alleles on the tops and the sides. Now, for me, it doesn't matter if you write the male on the top or the male on the side or the female on the top or the female on the side. That doesn't matter. You're going to get the same results anyway. So I'm not a real stickler about that. But you're going to write each allele on either the top or the side. So those represent the sperm and the egg segregating those alleles, separating those chromosomes, and then they're going to come back together in different forms in the offspring. So then what you're going to do is you're going to fill in the table. So, for example, that big B comes over into that first square, and then the top one comes down, and then that little one would come all the way over. The little b from the top comes all the way down. So eventually you would have all those squares filled in by bringing those down and across into the appropriate boxes. So once you've got the Punnett square filled out, then you need to take a look at the resulting phenotypes and genotypes. So this is where you would calculate either the percentage or the ratio of what they look like. So this, for example, we have big B, which ends up being homozygous for a large beak. Then we have those two squares that have the same genotypes. So big B, little b looks like heterozygous large beak. And then finally we have the two little b's, and that ends up being homozygous for a small beak. So our resulting percentages, we have 3 that are large beaks. So that's either 3/4 or 75%. And then we have 1/4 or 25% that would have a short beak. And then our ratio is 3 to 1, long to short. So for your percentages, I always just think of $1. So if it's 2 squares, it's 50 cents. If it's 3 squares, it's 75 cents, or that equals 75%. As with any rules, there are exceptions. Some of those to Mendel's principles are that some alleles are neither dominant nor recessive. Many genes exist in several different forms, and then many traits are produced by the interaction of several genes, and that's what we'll talk about next. So first we have codominance. Codominance is when both alleles are clearly expressed. So I always think of the prefix co means to share. So both of those traits are going to share the stage. You'll be able to clearly see them both expressed. And the example we have here is a roan cow. Roan cattle are a result of a cross between a red cow and a white cow. And you can see on the bottom in that Punnett square, the way that that's written is with 2 capital letters, because neither one of them is recessive, and they're both going to show because it's co. So we have the red cow is written as capital R, and then we have the white cow written as capital W. So when those alleles come back together, it's a capital R and a capital W. So that lets you know that they're both going to show. And there's a picture of a real cow there on the right. Incomplete dominance is when neither is completely dominant, but those traits result in a blending of the traits. And the example here is going to be a gray mouse. So that color would be a result of a white mouse and a black mouse mating and having offspring that were gray. Then we have multiple alleles. A single gene can have many possible alleles, and the blood groups are an example of that. So you can see in that chart on the bottom, we have group A, which is blood type A, group B, blood type B, type AB, and type O. So there's a little graphic of what those red cells kind of look like. And the reason that they're typed like that, AB, AB, and O are because of antigens that are produced. So red blood cells have proteins on the surface called immunoglobulin. That immunoglobulin produces antigens. Blood type is determined by the type of antigen that's produced. So type A produces A antigens, type B produces B antigens, and so on. Now, the way that the blood types are written is important. And you can see that there are multiple expressions of type A blood. So we have the phenotype, which is type A. This is in the chart at the bottom of the screen. Then we have the genotype. So it's written IA, or IA little I. You're going to have to write these, and we're going to practice this in class. But the I is used because of the immunoglobulin. And then the blood type is going to be that superscript there. So either A, B, type AB has only one genotype, and then type O, which is recessive, is going to only have one genotype. So we'll practice this more in class in the next week. Then finally we have dihybrid crosses. Dihybrid crosses is when we're going to track two traits at the same time. And that's similar to what you saw when we looked at the principle of segregation. Those alleles separate together. They don't necessarily fit together and go with each other all the time. So with two alleles for each parent, there's going to be 16 squares. And this is, again, something that we're going to practice in class. So this is to track two traits at the same time. And I know you use this acronym in class. We'll use it here for dihybrid crosses. So if you have the genotypes there, you can see those genotypes in that third column. So what you'll do is you'll do first, outside, inside, last. So you take the first genotype, then you do the outside letters, then you do the inside letters, and then you do the last letters of each pair. And again, this is something that we'll practice in class, so don't stress out about it too much right now.
I have a pen. I have a pen. I have an apple. Apple pen. I have a pen. I have pineapple. Pineapple pen. Apple pen. Pineapple pen. Pen pineapple apple pen. Pen pineapple apple pen.